So my talk is going to be very detailed and low level. Now that you've seen the top level view that Scott did, I'm going to be talking about how to get it done, basically. How to annotate a data set from start to finish with HED, but also considerations about things like event file design and some other practical considerations. And after I'm done, then Young and Monique will talk about some of the tools that actually enable this in a reasonable way in more detail. And my talk is going to be in 4 parts. The first part is I'm going to go through how annotation of events actually works in BIDS and try to give you a low-level understanding of how it works. Then I'm going to talk about the basic HED syntax and vocabulary, not in a lot of detail, but just to go through it again, as the things that Scott introduced. I'll go through a quick start on how to do HED tagging and emphasize that tagging is an iterative process and that you can start simple, and as you get a better understanding of what you want to accomplish with the tagging, you can put in more details, and this is easy to do by the way that it's organized. And finally, I'm going to go through some of the HED's more advanced features and how to use them and what the consequences of using them are in analysis. So we're concerned about how to analyze data in a way that minimizes the amount of code that has to be written that's custom code. So as Scott mentioned, we need annotations and data organization that's human understandable, but that's machine actionable, that you don't have to do some particular code analysis for each particular data set that you want to use. As Scott mentioned, all the tools and examples are online, and there's a number of links throughout this, but if you go to the GitHub standard, you can see links to everything there. The HED standard on GitHub. Okay, so let's get started with how to do it. And so first we have to understand how events are organized in BIDS, and for some of you this will be really old stuff, and some of you who are unfamiliar with BIDS, this will be new. But basically, an events file is a tab separated value file, and it has a column header. Each row represents an event marker, and the onset gives the time in seconds to the neuroimaging file that the events are linked to. So, and they're linked in BIDS by their file names. So there's a rule for, if you see an events file with a certain name, you can find the neuroimaging file that it goes with. Now, each column in the events file contains information about that event, and BIDS only really requires the onset and duration columns. And as Scott mentioned, a lot of data sets, most data sets ignore the duration columns. And BIDS really doesn't impose any requirements on what's in the events files, except that you have to have the onsets that correspond to the neuroimaging file. So you can add other columns as desired. And we think this is really important for understanding, having people use the data in a way that's accurate and not requiring a huge amount of extra coding. So the example that we see here is the Sternberg working memory data set, which we'll go into as one of the examples in this talk. And it uses a value column as the event type, and that's a value column is one of those mentioned as a recommended or optional column in BIDS. There's some other columns that are also mentioned in the spec, such as the trial type and the stimuli column. One of the unfortunate things about the sort of the backstory of BIDS is that a lot of the events, because the trial type is mentioned as a recommended file, a lot of data sets have each event not represent an event, but a single trial. And so encoded in there are all sorts of things like offsets for queues and offsets for response times, and all on a single line. In order to understand how those things occur relative to the anchor onset, requires a really complicated, careful reading of the documentation, which is often not there. And it also promotes not reporting events. And this is really important for EEG, but it's also important for fMRI. And so we highly recommend that you don't try to include all of the information about a single trial in one event marker, and that all the events be reported. They can always be filtered out later if you don't want them. So let's take a look at some actual examples. The one we just looked at is from the Sternberg, as I mentioned. This is a data set from Julie Anton and Scott. And it's been selected as one of the replication studies for EEG many labs. Each event file has about 350 events, and all of the events are encoded in the value column. And we'll get into some of the issues with that in the next couple of slides. But let me just mention that one of the problems is that if you that the clicks which are the button press responses, are encoded both for response and for saying get ready, and we'll talk about why that is an issue. Overall, this is a relatively clean data set as far as annotation goes. A second example is the audio visual attention shift data, and this is about 6,000 events per file. And this is also from Scott's lab. It's an early data set, and it uses two columns for encoding, an event code and a condition code. The difficulty with this encoding is that the event codes often mean the opposite depending on what the condition code is. And that kind of encoding makes it difficult for downstream tools to automatically annotate it or use it for analysis. And it's just asking for bugs and misuse. So, a recoding of that, we did do a recoding of that to get rid of that. But careful thought about the event coding is always important. Third example is from the BCI driving corpus. This file is about 3,000 events, and it's part of a larger corpus of about 12 studies from different institutions using different headsets and different paradigms. And they wanted to encode their events in sort of a common namespace. So they came up with a 4-digit encoding scheme, and the positions of the digits had some general class of meanings, and they agreed on all the different codes so that a given data set always used codes for the same meaning, which was great for analysis, but you had to have the magic spreadsheet in order to know what those codes actually did. Then after that program ended and another program occurred, they added some more data sets to this, and including these rapid serial visual presentation data sets. This data set, the event files, had about 20,000 events, and they wanted to use the same coding, but unfortunately all the codes were used up. So they had to come up with a 5-digit encoding scheme in order to be compatible with the other data sets. So this kind of gives you an idea of what the problems are with having these custom code books and what the implications are for downstream analysis, because all of these are going to require custom coding in order to do analysis. So let's look at the problem of ambiguous encoding, and we'll look at the Sternberg data set for that, which is one that we use in it. It's a very nice data set, so we always use it in our examples. So the sequence of events in this data set, a subject sees a fixation cross and then is presented with a series of 8 letters. The ones in green are ones they're supposed to forget. The ones in black they are supposed to remember. Then they see a minus sign, which is their working memory period. And then they get a probe letter. And the probe letter is shown in red. If the probe letter was one of the black letters, then they're supposed to press the right button. But if the probe letter is a black letter, then they're supposed to press the left button. So a right click means it was there, I remember it. The left click means it was not there. But the problem is that they also use the right and left clicks to respond to the to say they were ready after the feedback. And in theory, you know, that there's really nothing wrong with that, in that if you write code, you can filter those out. But things are always more complicated in practice than they are on paper. And in this data set, you know, there were some participants who enthusiastically double pressed buttons and pressed 2 buttons before the feedback, and there were some that the feedback didn't occur. And this is something that happens all the time in experiments. Participants don't exactly follow the paradigm, and that makes ambiguous encoding a little more difficult to handle, because you have to go through and allow for those ambiguities when you write your scripts, and it's very easy to get it wrong. So a better alternative is to have the button presses after the feedback be 3s and 4s rather than 1s and 2s if you're going to use that kind of thing. And that solves the problem. It's fairly easy for the data distributor to do, and will reduce errors downstream. So that's the idea of careful thinking about events and how you report them to reduce ambiguity. So another issue, which, well, this was the original encoding, but another issue is you want to encode the experimental conditions and possibly the task context as well as the stimulus. And this original encoding does a good job of encoding the stimulus and the response. Now, it's true that you have to split the G's out in order to know whether it's a letter to forget when you do the analysis. So there's going to be custom coding there, but it is mostly unambiguous. But it doesn't really encode things like the experimental condition or the task context. And so a data distributor who carefully thinks about what to present can produce an event file that's much more informative and much less likely to be misinterpreted. So we recoded this and we used sort of an orthogonal encoding. We like to, this isn't required, but it's useful to separate the idea of what happened in the event, that's using the event type, and what that meant in the experiment, which we put in a task row column. That the column names are, you can do whatever you want, but you know, this is as good as any. And then some specific things like what letter, we put that in a separate column, and that can be annotated separately. We also have found that it's really useful to put trial numbers in if it's a trial-based experiment, because all experiments have little failures and glitches, which make it difficult to just have a uniform way of determining where the trials end, which events are in the trial. And by doing that, when you distribute the data, you greatly reduce the chance that someone will misinterpret it. And so we really like to devote a column number to that. Now, so the goal here is to make event files that are clear, that they can be read and understood what the events mean, and that will have annotations associated with them that can be directly used by the analysis tools without a lot of custom coding, and ideally no custom coding. It can be used across data sets so that to enable cross-study comparisons. So that's our goal. Sorry. So now I'm going to get down to how event annotation is done in BIDS using HED. Excuse me just a second. And Scott introduced the idea in his talk. So hierarchical event descriptors, they are fully integrated into BIDS, and they're validated as part of the BIDS validation. And there are two ways to put HED annotations into BIDS. One of them is direct, and the other is with JSON sidecars. Now, direct annotation, you put a head column in the events file, and you annotate each event separately. And head is one of the optional columns specifically mentioned in BIDS, but annotating the individual events is not a very attractive option. It's hard to maintain. Think about those event files with 20,000 events in them. If you want to change an event or make sure that the annotations are consistent, you have to write a bunch of scripts, which are unpleasant at the least and probably wrong at the worst. But luckily, there's a lot easier way to do that, and that is with JSON sidecars. So here's how the JSON sidecar annotation strategy goes in BIDS. I'm going to start on the left with the Sternberg Wake working memory excerpt event file. And instead of trying to annotate each individual event in here, which will be thousands of annotations, I create a dictionary of the unique values in the column. And usually that's a much smaller number of annotations to do. And those values are typically common across the data set. So, it's typically 10 to 100 values, and they're shared across the event files in the data set. And so I create the dictionary and I fill it in with the head annotations for each entry. Once I've done that, I'm done basically for a head annotation of a BIDS data set. Now I've shown the data set as a table, but in reality, it's a JSON text file, but you have the option, we have head tools which will allow you to annotate in a spreadsheet and convert to a JSON sidecar, or do it directly in the JSON sidecar. So if you like doing a table, which I prefer to do, you can convert it directly to a JSON sidecar. So at this point, you're done with your annotation once you fill in that file. Then at analysis time, there are tools available which will take the events file and the JSON sidecars. And if you need to put head columns in your event files, sometimes you need to, it'll assemble all of those annotations to create a single head annotation for each event in the data set, but that's only done at analysis time. At an annotation time, you're just concentrating on expressing what those events mean. So then once you, at analysis time, you can filter on head tags and select tags for epoching data for ERP analysis if you're doing EEG. You can create factor vectors for doing the SPM analysis that is typical in fMRI, but you do it based on tags rather than on writing code. And if you filter, then you can reduce your event file, or you can put factor vectors as columns on your event files in order to do a linear analysis. The options are open. The head tags can also be used for searching, and we have a number of tools under development for summarization of data sets in compact form, which we hope to incorporate in some of the archives, such as NEMAR, which is in Scott's lab. So let's look at this in a little bit, how this actually looks in a JSON file. So the JSON file that might be used to annotate the dictionary we saw here, this is what it looks like. Each column in the spreadsheet that you're going to annotate is a key in this dictionary. And BIDS has in its, without head, you use levels to describe what the items in the dictionary mean. And you can use the levels with the head tags. Ideally, you would use both. And when you assemble event annotations for analysis, you have the option of including the level descriptions as part of the annotation as a description for searching. So this is what the event table looks like. You fill in those head tags, and then you save your JSON file. And usually the JSON file is applicable to all of the event files in the data set. So you only have to put one JSON file at the top level of your BIDS data set. If you want to go back and change your annotations later, you just have to edit that data set, that JSON file. You don't have to change anything about your events. So that's the process. You get a JSON file or a table with unique values. You then annotate those values using head, and you put the JSON file in the top level of your BIDS data set. So the idea is that you annotate each unique column only once, and you have a single, ideally, you have a JSON file that applies to everything. Now, you're allowed to have other JSON files if there are some specific subjects that have special things in them, and they all apply using the BIDS inheritance principle, but I won't get into that in this talk. Now, you might be wondering at this point, okay, well, I have to now write code to find the unique values in my event files, but actually you don't, because we have tools for that. So there are some online tools available at head tools UCSD.edu/head. And they make the setup of the JSON annotations a lot easier. So if you go there and select the events option, you'll see a menu like this in the online tools, and there's some actions you can pick. If you want to try out your head annotations that you've done in your JSON file with an events file, you can do a validation and it'll check them. And that's an easy, fast way to get the annotations correct. If you want to see what it looks like to assemble them, there's an assemble option. But the thing we're interested in here is to generate a sidecar template. And so you just select an events file. And when you browse to the events file, then it'll change the menu to bring up the following. So what the tool does is it looks at the events file and it tells you what the contents are, what each column is named, and how many entries, unique entries, are in each column. And so you then select the columns that you're going to include in the annotation. And usually you don't want to annotate the onset at the duration of the sample, but you could. Usually you don't. And you check those on the left in that menu. And you then select which columns are going to be treated as having sort of being categorical columns. BIDS calls those ones that you put levels in. And they allow other columns, which we'll call value columns, which have non-categorical values like response times or things like that. And HED has a convenient way of annotating those as well. But they're not annotated as categorical columns. You select those columns, you press process, and it generates a template for you with all the unique values and levels and dummy entries for you to edit. So you'll notice that it doesn't have up here, let's see. If you look at the tags there, they're dummy tags. They're just labeled. This is a valid head. Label is a valid head tag. And it just puts in a label for the column name and for the column value. And then you just edit those with what you want to do as an annotation. And hopefully you also just do the descriptions as well. Now, I just like to point out the letter column, or the letter column, which is a value column. We're going to annotate that as a value column. And in letter columns, you just, instead of annotating each unique value that might appear in that column, you just annotate the column. And you do that by putting the head in a head category under the column name. And you, somewhere in the annotation, you use a number sign. And the number sign replaces the column value, is replaced by the column value in the annotation. So in this way, you can annotate all those values, let's say response time, using just a single annotation. And when they're assembled, it works. So let's just sort of stop. This is sort of the first part of the talk, which is sort of the how to do it. And let's just step back for a minute and look at what we've talked about. So I think the key points are that events are really essential, and it's important to pay attention. One of the things that we find extremely frustrating as we're working with data is that if events aren't reported, they can't be used. And usually if they're not reported, they're not going to be recoverable either. So they're gone. And that precludes using a lot of data for for analyses that maybe the original data authors didn't anticipate, and it's an opportunity lost. So reporting events is really important. It's also important to have well-structured event files and unambiguous event codes. If you don't, someone who uses your data is going to have to write code to reorganize it or do other kinds of custom coding. And in a lot of cases, just a little bit extra work to make the files well structured will save a lot of work downstream and really expand the use of the data. So identification of what the codes mean, regardless of what kind of codes you use, is really important for correct analysis. And believe me, it's really hard to get it from the papers. BIDS has an easy mechanism for doing event annotation, just using this single top level JSON file. And for a lot of annotations, it's the matter of just producing 20 lines of annotation to annotate the whole data set. And once you get into it, it becomes much easier. So there are quite a few HED tools available, tools to assemble the data, the event annotations at analysis time, and a number of other tools for creating factor vectors and extracting experimental designs and other things. Later in the workshop, Young is going to talk about some tools for annotating and analyzing data using HED. And Monique is going to discuss our new files restructuring tools, which will help in producing those original event files or adapting event files downstream. So next up, I'm just going to quickly go through the basic HED syntax and vocabulary. So Scott has gone through this already, but let me just review it. HED annotations are comma-separated lists of head tags. They can be parenthesized. We strongly recommend that you use the short form. It's easier to read. And the tools take care of everything behind the scenes, so don't make extra work for yourself or other people. As a result, the being able to use these terms in short form required some redesign of the vocabulary from HED 2 to HED 3. In particular, the obvious thing is that all the terms have to be unique, but a more important, well, not a more important, but an equally important thing is that the terms have to stand on their own. So terms that might make sense if you put a full path, but don't stand on their own, would not make it into a HED library or the HED schema. So as had Scott mentioned, the HED tags come from a specific vocabulary, which is available on GitHub, and the tools can automatically download it from GitHub if you use it if you just give the HED version. So there are officially released HED versions that should be used. You are allowed to extend those official versions, but one of the points of having a standardized vocabulary is to be able to do analysis across data sets. And if everyone makes up their own vocabulary, that's less likely to happen. But the tools can handle any vocabulary that follows the syntax and the rules. So as mentioned, you can reference a tag by path or by the individual node name. Usually we want to just do it by the node name. An important requirement of the HED hierarchy is that the terms under a given term satisfy the ISA. That is, a sensory event is an event. And this allows you to search, to do search generalization. So if you search for event, you can get all of the tags that are anywhere in the subtree for event, and similarly for other action. So each head term stands on its own, and it satisfies the requirement that it is a type of the thing that was its parent. Oh, sorry. And the view, this is a viewer on the left. It's available. You can look at all the head tags online. I got a little bit ahead of myself here. So there are some other things about the head hierarchy that I just want to mention. There is a structure. There are certain top level trees that are noun-like, some that are verb-like, and some that are descriptive. The event, agent, and item are noun-like. Actions, verb-like, and property and relation are descriptive. And this allows you to sort of form pseudo sentences, which are parenthesized groups of head tags that have a noun, verb, and a direct object phase. And we currently don't have tools that handle that, but we plan on having some things that take advantage of this for more advanced kinds of reasoning downstream. So now we get down to it, which is part 3, which is how to actually put head tags in and how to pick them. And recall that our task is to fill in a dictionary, assigning head tags to the unique values in the event file. And what we're going to do is give you a recipe, which is what kind of event. And then once you've selected what kind of event it is, how you add supporting tags. And we actually have an online, in our online tutorials, the head annotation quick start, we have a table which will allow you to do a basic head annotation fairly quickly. So the first thing you do is you figure out what kind of event you have. And if you do no other annotation and you at least have what kind of event you have, you're providing something useful. So figuring out the kinds of events is probably the most important thing for downstream analysis. The second most important thing I'll just say right now is knowing what the experimental conditions are and being able to put those in, but we'll get to that a little later. So you figure out what kind of event you have, and then you want to be able to add additional tags of description. And it's an iterative process. So you can start simple and add more tags later as you figure out more details. But there's some basic things that we want to do to just about everything to do basic annotation. So let's just take a look at sensory event, which is, typically experimental stimuli, but it can also be other things like cues and feedback and other things. So if it's a sensory event, you can't assume that it's just a stimuli. It could be lots of other things. And it's important to have those identified in the experiment because you want to be able to regress out the effects of these. Even if you're just interested in stimuli, you want to be able to regress out the effects of the other stimuli when looking at your responses. And so you want to have all the events in if you can. And some of them will be task related and some of them will not be. So you want to say what kind of sense was used. Is it an auditory presentation or is it a visual presentation? Then you want to talk about what its task role is. Is it instruction or is it a stimuli? And if it's a stimuli, what its purposes. And if it's and once you get that down, you probably want to say something about what was being presented, and that could be, you can really go off the deep end and do lots of detail, but at least put something in. And so if it's a face or it's a sound like a siren, that's useful. And so this table has sort of this recipe where you go through and you pick your event, and then you say, okay, here's my checklist, put these tags in. And once you've done this once or twice, you essentially are familiar with it, and you know what you need to put in for a basic annotation. So if you look at these example, this non-working memory is the fixation, no, it's the dash at the after the letters are presented in the Sternberg data. And it's a sensory event, it's a visual presentation, its purpose is instructional, it says get ready, and it's a cue, and it's, the thing is, it's a dash and it's black. So that's an example. And here's another one, which is the feedback. It's an auditory presentation and it's a beep. So this is what a basic annotation in HED looks like, and you fill those into the dictionary. Now, the next thing I want to show you is how this all gets put together at analysis time to produce an annotation that can be used by the computer. So we start with an events file. So this is one of the lines in that Sternberg events file. And my example, I'm going to annotate the event type, the task role, and the letter. I'm going to skip the memory condition. So in the JSON file, under the event type key, they're going to be the annotations for the things that can appear in the event type column, and show letters looked up. It's a sensory event, visual presentation, character, and it's in the center of the screen. And then the task role is also looked up. It's another key in that JSON file, and there are a lot more task roles in there. And then the letter column, I'm going to annotate as a value column. And so the key there is it's just one head annotation, which has a number sign that's substituted. The column content substitutes for the number sign. And so once those are looked up, the assemble tools will create a head assembled head tag for that particular event. And this isn't done and put in the event file. It's put, it's done at analysis time on the fly. And it's a fairly fast process. Okay, so here are the key points that we've covered so far. Head tags are paths. They're in a hierarchical event structure. The tags are unique, and so you just want to annotate with the single node name, not the full path. The head vocabulary follows the ISA, so you can do searches for terms higher in the tree, and that will return all the tags below it in the annotation. Head is well integrated into BIDS, and if you're going to use head annotation, which hopefully you will, the preferred method is to use a single top-level sidecar containing the annotations. But you can use sidecars at multiple levels if necessary, and you can annotate individual events in the head column, and the assemble tools take it all into account to produce a complete annotation. So finally, it's hard to come up with the perfect annotation for all purposes. And I think it's important to recognize that annotation is an iterative process. And it's it's best to start simple and then improve your annotations as you come to understand more about your data. I have one final section in the talk, which is the more advanced features of HED and how to use them and what their implications are. So let me now turn to that. And I'm going to use the Wakeman-Henson face processing data set that Scott introduced in his talk to illustrate that. And so you've already seen the experimental timeline that Scott showed you. So the sequence of events within a trial, the participant sees a fixation cross, they're told to fixate, not blink. They see a face, and then they press either a left press or a right press, depending on whether they view the face as being more symmetric than average or less symmetric than average. The symmetry evaluation task is a sham task. But one of the other problems is if it weren't a sham task, and some people may be interested in the symmetry evaluation and how it corresponds to some of the other behavioral data. So it shouldn't automatically be treated as a sham task. But one of the problems with this, with the encoding, is that the key assign that whether a left press means more symmetric or a left press means less symmetric is dependent on the experimental condition. So this is another one of those, the condition controls what the other codes mean, and it's the opposite depending on what the condition is. So when that kind of a coding appears, you have to be really careful because it's very easy to get them reversed. So the data set has 3 experimental conditions: the type of face, it is, which had 3 items, a repetition status, which was whether the face you saw last is the first time you saw that face, whether it's the one you saw just in the previous trial, or whether it's one you saw quite a while ago. So there are 3 cases there. So that forms a 3x3 design. And then there's this other condition, which is the left button means symmetric, or the right button means symmetric. So we're going to now look at how to use HED to encode this information. And I'm going to concentrate on the face type, but let me just point out that the face type and the repetition were in columns in our encoding, and whether the left click means more symmetric or the left click means less symmetric is encoded by a single event at the beginning of the file. And this encoding was done because that experimental condition was varied by file rather than by trial. The other 2 experimental conditions, the type of face and the repetition status, changed with each trial. And so those makes more sense to encode in the columns. Okay, so let's look at how HED encodes experimental conditions. There are 2 ways to do it. There's direct and there's defined. And so here we're going to encode the face type, which is it's a column of things. And to encode the face type using the direct method as a condition variable is really simple. You just say condition variable, and then you name it, and you can name it whatever you want, and usually you name it something that resembles your experimental conditions. The direct way of doing encoding of condition variables is easy, and it is included in the summaries. And you can have the tools, which we have, create factor vectors. So you can get a column with ones where it's there and zeros where it's not. And that's useful for linear modeling. But it has some disadvantages. And in particular, it doesn't allow you to extract directly the design matrices. And you can't really say with this what those conditions mean. And so there's another way of doing this with the defined, which does allow that. And I'm going to talk about that next. So in defined, instead of saying condition variable when you're whatever you want, you say def and whatever you want. And just using the def means this is a defined term, doesn't necessarily say it's a condition variable. So it can be used for lots of things like defining particular things, a meaning in the lab or whatever. What makes it a condition variable is, and if you use the def tag, you have to create a definition somewhere, which you can put in the JSON file in a dummy column, which I'm showing here. But when you do the definition, you have to put in the definition that it's one of the tags, that it is a condition variable, and associate a name with that condition variable. Then the def tag is treated as a level for the condition variable. So famous face condition is treated as a level of the condition variable face type, and you have a design matrix there. It has this has lots of advantages because it allows you to do design matrices, and you can put in, you can describe what those conditions are and use that in analysis also. And definitions allow you to define events of temporal extent, so things with onsets and offsets and phases. So I'm now going to show you, make a comparison of those two. So in direct, you just use the condition variable tag and reports like this. And we have automatic summary tools that will summarize a data set from the JSON files and whatever head tags you provide in there. And this is what the summary for condition variables looks like for, this is for that data set. So if you just the usage of condition variable and famous face condition automatically allows you to extract the summary and identify this as a condition variable. And this says that, for example, unfamiliar face condition appears in 5,320 events, and there are a total of 63,320 events in the file, and so on. So you can get the automatic summaries, but it doesn't relate any of these three as being varying of levels of a single condition variable. So you miss the design matrix aspect of this. With defined, instead of the condition variable, you use the def tag, and you have to give the meaning of the tags in some definition. But when you do the automatic summary, it's able to recognize that unfamiliar face condition is a level of the condition variable face type. And this is, remember, this is done automatically. You just run this and you can get it. So again, we have 63,320 events, and unfamiliar face condition has 5,320 events. So you get the same counts. It appears in 108 files in the data set, but you now have the design matrix extracted from this. So let me now just do the temporal extent. So we're going to try to encode this one. So the idea with direct is you again just use that, and if you look at the summary, the problem is you'll see that that event appears in 60 times in the data set, but there's 60 files, so it only appears once, and there's nothing there that tells the analysis tools that that setup left symmetry should apply to the entire data set. With defined events, you do your def and you just say it's an onset. And that then tells the tools that that event continues on until it hits an offset or the end of the file. When you do the summary for that event, you still get this, the same number of total events, but it shows that there are now 28,000 left symmetry condition events corresponding to the 48 files that use left symmetry condition as the event. So that's the difference between those. So let me just step back a minute and just give review. So the points of this presentation were to try to convince you that events are really central to an analysis and that it pays to do a careful handling of events before distributing data on a site like OpenNeural. It's also important for your own analysis and can really reduce the amount of code that has to be written downstream to handle the analysis. So HED is a general framework. There are a number of tools as well as the annotation vocabulary. But another important concept that I want to leave you with is that it's really impossible to anticipate all the forms that an event file will take. And so, or what will be needed for future analysis. So we'd really like to promote the view of event files as having fluidity. I mean, you have to report the events, but how you represent them is really open, and you may need one kind of representation for one application and another for a different one. So reporting the events and reporting them unambiguously is really important, but the format can be transformed later if you have different needs. And there are tools for restructuring event files and for searching and summarizing using head tags, as well as for things like producing design matrices and factor vectors for analysis. And there are some links on GitHub, which I would recommend that you take. We are going to, let me just, so there are a bunch of links, which I'll just mention here. We're going to be taking a bio break after my talk, and after we come back, and Scott will jump in and tell us when we're coming back, Young will present, actually do some demos of some of the tools that are available. There for the accessing the web services, some of the EEG Lab plugins for using HED, and the standalone CTagger for doing HED annotation with a GUI. And Monique will talk about some of the tools that have been recently developed for event file restructuring, which is important both for just creating the event files in the first place and doing the analysis downstream. And then Dora will be presenting our exciting new score library for EEG clinical neurological annotation at the end.